So what's inside the FASTQ file? So FASTQ files are basically just plain text files that you could open on your computer in a text editor, but they are very big, so it's not always advised to do this. And they contain the reads. That's the sequences that you see here and here. And additionally, they also contain the quality scores, and that's the things that you see here and here. So quality scores that were generated during the base calling. So what are reads? So reads are the actual sequences that are generated by the next-gen sequencing process. Every polony or every cluster in case of Illumina data represents one fragment from the library. And after sequencing, each of these polonies or each of these clusters will give rise to one sequence, and that sequence is called a read. Reads are much shorter than the fragments they originate from. So fragments are typically between 100 and 900 nucleotides, while reads are between 40 and 150 nucleotides. So a read is just a snapshot of one end of the fragment. In single-end sequencing, every fragment will generate one read, and in paired-end sequencing, every fragment will generate two reads. So in single-end sequencing, only one end of the fragments is sequenced, thus generating one read per fragment. In paired-end sequencing, you actually do two sequencing runs, and in each sequencing run, you sequence another end of the fragment. So every fragment will generate two reads coming from its two ends. So reads consist of bases. That is what you see here. So the bases are the letters that you see here in the sequence, the A, the T, the C, and the G. They represent nucleotides. So in each cycle of the sequencing, one nucleotide is added to the growing strand, and the identity of that nucleotide, whether it's an A, a C, a G, or a T, is determined by the base calling software. So every base is literally one letter in the read. So this is what a FASTQ file actually looks like. So as I said, it's a regular text file where each read takes up 4 lines in the file. So these first 4 lines, they belong to the first read that was sequenced, and the next 4 lines, they belong to the second read that was sequenced, and so on. The first line is the read ID. That's what you see here. It's a unique name for each read, and it tells you where on the flow cell that read comes from. So the only thing you know at this point about each read are the sequence of the read and its location. So where was the corresponding polony or the corresponding cluster located on the flow cell? The sequence is not necessarily unique. So FASTQ files contain identical reads, and we will come back later on this. But the location of every cluster or every polony is unique for each read. So it's the only thing that you can use at this point as a unique identifier for the reads. And this ID is built up as you can see here. So first you have the machine ID. So the ID of the sequencer on which the sequencing was done, the ID of that specific run in which the read was sequenced, and then you have the actual position, so the number of the lane, the number of the tile, and then the X and the Y position of the cluster or the polony. So in this case, we're talking about the cluster because it's Illumina data, and this read was located on lane number 1, tile number 1, and in this position on the flow cell. FASTQ files from Aviti and Illumina look the same. As you can see here, they have the same format with 4 lines for every read. This is also the case here in this Aviti FASTQ file. And they have the same format for read IDs. So also here you see that the ID is built up by first giving the ID of the sequencer, then giving the ID of the run in which the read was sequenced. In Aviti FASTQ files, they also add the ID of the flow cell, and then you get the location, so the lane, the tile, and the X and the Y position on the flow cell. The third line is not important at all. It's either a repeat of the first line or it's completely blank. The second line contains the actual sequence, the read, and the fourth line contains the quality scores. That's what you see here. So the quality scores tell you how confident the base calling software was about calling a base. So every base in the sequence has a matching quality score here in the fourth line. So the two lines are the same length, and this makes it easy to see, oh, for this T here, we have a quality score that is equal to a lowercase h. And for this T here, we have a quality score that's actually a capital T, and so on. And this is also the case in Aviti FASTQ files, as you can see here. So every base has a corresponding quality score.